Good morning, everyone. It is indeed a great pleasure to talk to you about my work on genomic aspects of Ayurveda Prakriti. In fact, I must thank Professor Akshay Anand and Sheetal Gupta for this initiative in this unprecedented time. I'm a geneticist. I mostly work on human genetics in various aspects, particularly try to understand how the modern humans have evolved, migrated, and populated around India and outside India. Another aspect of my work is understanding the genetic basis of several diseases, including cardiovascular disease, mitochondrial disorders, male infertility, and so on. I started the Ayurvedic work about 10 years back. In fact, that was initiated by Professor Valiyadan. Most of you are aware that he is a well-known cardiac surgeon, very keen in understanding the scientific basis of Ayurvedic Prakriti and many other aspects of Ayurveda. In fact, he organized a brainstorming meeting in the year 2006 at the Office of Principal Scientific Advisor to the Government of India, where he invited few scientists and Ayurvedic physicians to discuss and evolve projects to understand the scientific basis of Ayurveda. I was fortunate to be a part of that discussion, and I proposed to do genome-wide genetic study to understand is there any scientific basis or genetic basis for Ayurvedic classification or Prakriti classification. Some of you are aware there are three Prakritis, Vata, Pitta, and Kapha. As a geneticist, I believe that everything has a genetic basis. For example, we have mutation in gene which leads to several diseases, and we have mutation in some of the genes which gives us resistance against some of the infectious diseases. We have different kind of mutation which makes the individual to susceptible against some of the pathogen. Same way, we have mutation in pharmacogenomically important gene which gives a therapeutic effect. Some mutation may give adverse effect. Some mutation may not give any effect at all. On this basis, I thought that there should be definitely some genetic basis for this Ayurvedic classification. So I proposed to do genome-wide genetic study to understand how we can make use of the genetics to understand the Ayurvedic Prakriti classification, that means Vata, Pitta, and Kapha. So these are all based on certain phenotypic features, certain basis which the individual likes or dislike, and also based on the pulse. So this discussion has evolved to do genome-wide association study to understand the scientific basis of the Prakritis. I had two other collaborators, one from Manipal University, Professor Satyamurthy, who proposed to do the methylation analysis of these Prakritis, Professor Kondaya from Indian Institute of Science to do gene expression analysis. And we had three major Ayurveda institutes, one from Bangalore, one from Udupi, and another one from Pune. As some of you are aware, the Ayurveda classification is based on several questions, features, and so on. And none of us are with one particular prakriti. It's always the prakriti exists in the mixed form. For example, if I have been diagnosed with vata, I may have 60% of vata, 20% of pitta, and remaining 20% of kapha. Or somebody may have 70% of vata, 20% of pitta, and 10% of kapha. And what we decided to have is somebody who have 60% of one particular predominant prakriti, and we consider that the individual has that particular prakriti. So our Ayurvedic clinicians have screened more than 3,000 individuals. Our ultimate aim is to have 100 individuals from each prakriti. And they also had very well defined inclusion criteria. They included only the males. They don't want to have too much of confounding effect. So males with age between 20 and 30 years, who are apparently normal, doesn't have any other well-known disease history. After screening, they have been reassessed by computer assisted technique, and those individuals who had concordance by both Ayurvedic physician and computer assisted questionnaires have been included, and those who have one particular prakriti with predominant, more than 60% of one particular prakriti has been selected. We end up with identifying 100 individuals with one particular predominant prakriti, that means 300 individuals. All of them have been subjected to 1 million analysis of 1 million genetic markers using Affymetrix array, which consists of 1 million single nucleotide polymorphic markers. We used to use these markers to understand the genetic history of each population. These markers are distributed all over the genome. And if you take, for example, some 100 Indian population who are ethnically very unique, then you would expect to see the 100 cluster when you do principal component analysis, because each population has very unique set of mutation. Therefore, all of them have been clustered independently. So when we analyze 300 individuals with 1 million SNP markers, and we used different statistical analysis, one of the analysis is called principal component analysis. When we used these 300 individuals, 100 from each prakriti, vata, pitta, and kapha, we have seen there's no pattern in the cluster. They're all distributed all over the PCA, principal component analysis plot. That is expected because the samples are randomly collected. Now the question is how to differentiate these three prakritis, because they are all normal individuals. when we do case control study, we use patients and controls, and when we analyze, because we have one set of samples with the patient, other set of sample with control, when we do this analysis, then we expect whatever the difference between them that emerges as a difference which attribute to the disease phenotype. So in this case, because they're all of normal individuals, and the challenge is how to differentiate them. Then we evolved a different method, taking one prakriti, individuals from one prakriti, that means vata, then compare that with other two prakritis, pitta and kapha. Now, whatever the difference we see in these 1 million SNPs, that means some markers, the frequency of which is very different in vata compared to pitta and kapha, are the one we have selected. Same way, if you take pitta, then vata and kapha has been compared and selected the markers which is showing difference between pitta and other two prakritis. Same way, kapha versus vata and pitta. After doing this analysis with extensive statistical analysis, 1 million times we have done permutation computation and picked up 52 SNPs. Then we did principal component analysis and found now there are 3 different clusters have emerged. While we doing the principal component analysis without any analysis, just putting all the 1 million markers from 300 individuals, we didn't see any pattern because these are all random sample. After this analysis, now we see clearly there are 3 clusters. Vata one cluster, pitta one cluster, and kapha as another cluster. Then we continue to do to see is there any genotype and phenotype correlation. Although these markers are from non-coding regions, we picked up very significantly where there's a gene called PGM1 which is involved in metabolism, and this gene is predominantly associated with pitta prakriti, and pitta prakritis are known to have high metabolic rate. And this additional evidence suggests that there's a definite scientific basis for Ayurvedic classification. So our ancestors who have developed and practiced for several thousands of years is having a definite scientific basis for this classification. In modern terms, what we say pharmacogenomics is where the genes which are involved in metabolizing the drugs, as a mutation which determines whether the drug is having a therapeutic effect or no effect at all or adverse effect. Same way, our ancestors who have developed the Ayurvedic principles and start treating the individuals based on the prakriti, one can consider that this is an ancient form of pharmacogenomics. So all this suggests that Ayurveda is not simply being practiced over a period of thousands of years, but it has a definite scientific basis. So I must thank Professor Valiyathan who has who initiated this, and later he formed a task force in DST to have Ayurvedic biology. Also must thank all our Ayurvedic collaborators, Professor Satyamurthy and Professor Kondaya, who were part of the study, who has done um, gene expression analysis and methylation analysis. And of course, the funding has come from DST. And thank you very much to all of you for listening this. Thank you.